Okay, before we start today's chapter, any questions for me from any of the previous chapters, what we did last week? So last week we got introduced to basics of human genetics, and we ended the class with Mendelism. We learned about Mendel's laws. We learned that he was a very good scientist, a very good record keeper. He was, his experiments were statistically sound, but he was also very fortunate because the plant that he studied was a sexually reproducing plant, and the traits that he selected from that plant were all controlled by a single gene. Each one was being controlled by a single gene. So those were single gene inheritance patterns. But today we are going to learn about deviations from Mendel's laws, because most of the genes in human beings are not controlled by a single gene. So you will be learning a lot of new terms, each of which is a deviation, is a different kind of deviation from Mendel's laws. But as you will go into details, in higher studies or whatever, when you learn about all these deviations, you see that these different terms, they start to blur and they start to kind of overlap with each other. So remember that in the real world, today's world, we do see most of the inheritance patterns are actually exceptions to Mendel's laws, because as per Mendel, each trait, for example, plant height or flower color, had two distinct forms, but most genes would not display such simple inheritance patterns. But one thing you have to remember is whatever Mendel predicted, genotypically, always the ratios persist. It's just the variation in phenotype. So externally, we may not be seeing what Mendel had reported, but if you go to the genetic constitution, genotype wise, everything still holds true. It's still true. So there are a lot of outside the gene influences which cause these deviations. For example, in human population, if a mutation occurs, okay, and sometimes what happens is that mutant allele, if it is dominant, okay, if it's not a recessive trait, if it's a dominant trait, then if an individual, if an embryo gets a double dose of that mutant dominant allele, that embryo may not even survive. So you won't see any live births with that particular genetic constitution. So that means a total class among the progeny is gone even before birth. But if you looked at what was the probability and calculated the genotypic ratios, yes, that embryo was formed, but that embryo got spontaneously aborted because the allelic combination, the genetic constitution made it lethal. We call it lethal alleles. And it's a classic example is a type of dwarfism. It's called achondroplastic dwarfism. So there are two alleles. Let's say small a, which is recessive, is normal height wild type, while the dominant mutation is a capital A, achondroplasia. This is what gives a person dwarfism. So if it's two capital A's, that means double dose of the dominant alleles, it becomes lethal. This individual will not survive. This will die in the embryonic stage. If it's a capital A and a small a, that means the heterozygous person is going to be born dwarf. Okay, he's a heterozygous person, and the recessive two small a's is of normal height. So if a dwarf, achondroplastic dwarf, marries another achondroplastic dwarf, so a capital A, small a, again a capital A, small a. This is what Mendel had predicted, 1 to 2 to 1. This is the genotypic ratio. You see this in the Punnett square. What happens is this particular box, the double A, these 1/4 of the embryos are going to die. They are never going to be born. So as soon as this fertilization occurs because of the lethal allele combination, this particular embryo will get aborted. So phenotypically, you will never see a double A, a capital A person, because it's lethal combination. Now, another deviation is not all genes would have just two versions, just two alleles. A gene can have multiple alleles, okay? That would show deviation. For example, there's a particular autosomal recessive disorder called phenylketonuria. These are biochemical disorders of metabolism. And this has hundreds of alleles, about more than 950 alleles, and which can give you a range of phenotypes from the mildest version to the most severe version. And the other allelic combinations would have symptoms lying in between. Or another autosomal recessive disorder is cystic fibrosis, and we've been using this example in other lectures also. This has over 1,500 alleles. So when you have multiple alleles, then you will not see that phenotype. And another kind of deviation is incomplete dominance. To understand incomplete dominance, let's look at this example first. This is easier. This is a particular type of plant called snapdragon, and the flowers come in many colors. You see some plants with red flowers. Okay, this is the dominant one, capital R, capital R. And then some flowers would have white color, a small r, small r. When you cross these two, ideally as per Mendel, phenotypically, this particular F1 generation should have all have been red because you have one capital R. But this capital R is not completely dominant over the small r. Okay, it is incompletely dominant. So when there is incomplete dominance, the hybrid shows a trait intermediate between the two. So the plants end up having pink flowers. So you see here, when you cross F1 to get F2, that means a capital R small r is crossed with another capital R small r. These are the different combinations of progeny that you get. You get some with red flowers, some with pink flowers. So this heterozygous becomes pink flower instead of red, and this is white. So this is an example of incomplete dominance. Now, in case of human beings, where do you see this? This is a particular gene that causes a problem called FH. FH stands for familial hypercholesterolemia. So what happens is, in the general population, okay, you have this particular gene is being expressed, and there are receptors. That means there are welcoming people in your liver that take the cholesterol from blood and detoxify it in your liver. Your liver is the detox center inside your body. So it keeps the level of cholesterol down in your blood. Now, people who are homozygotes for this particular mutation, they have very low number of receptors on their livers. So the liver is not efficiently taking the cholesterol from the blood. So these people will show a very high level of cholesterol in their blood. And when you look at heterozygotes, the people who are heterozygous for this gene, they have a level of cholesterol which is intermediate between the general population and the homozygotes. Another deviation is called co-dominance, means both alleles are equally, dominant. Nobody is dominating over the other. And a very important example here is our blood group types, We know that human beings, we have the ABO blood type. That means human beings can either be, you can either have a blood group that's A or B or AB or O. What does that mean? This means our RBCs, our red blood cells, we have antigens or proteins sticking out. And then there are two types of proteins or antigens, A or B. If a person has this A antigen sticking out of the red blood cell, then this person's blood group becomes A blood type. If the person has a B antigen sticking out, then this person's blood type is a B. And if a person has both A and B sticking out, then A and B are equally dominating. So the person's blood group becomes AB. Okay, it's not just A or not just B, it's AB type blood group. While if a person does not have any antigen sticking out, then this person's blood is O, type O. So when a person says that I'm A blood group or A blood type, genotypically we can write it as, this is the I, okay, for the allele is IA, if this is homozygous for A antigen, or it could be IA and a small I, which means there's nothing here, there's an O here. So this means this person is a heterozygous A. Similarly, a homozygous and a heterozygous B. For a person who has type AB, blood has to be IAIB, while a person with type O is two small i's. So this is what the blood grouping looks like, and you see A and B here are equally dominant, giving a person an AB blood type. So this is co-dominance. Next effect is epistasis. So epistasis means where one gene is affecting or masking the expression of, that means masking the phenotype of a second gene. And to understand this, let me first talk about the easiest example is red hair in human beings. We know that the hair color comes from pigments, and there are two main types of pigments, two types of melanin. One is called eumelanin, which is responsible for giving a dark color to the human hair or eyes. And there's another pigment called pheomelanin, which causes red hair. So depending on how much eumelanin you have in your hair, let's say if you do not have much eumelanin, your hair color becomes blonde. If you have intermediate levels, then your hair color is brown. If you have a lot of eumelanin, then your hair color becomes black. So this is just one gene controlling eumelanin. Now comes this epistatic effect of this pheomelanin. Pheomelanin is the pigment which usually gets converted into eumelanin because of this gene called MCR1. So MCR1 prevents the pheomelanin from expressing itself. So that is why redheadedness is kind of rare. You don't see as many red-haired people. It's because of the MCR1 gene converting the pheomelanin. But if there's a mutation in the MCR1 gene, then MCR1 gene does not work. And then pheomelanin expresses itself and the person has red hair. So this leads to redheadedness in human population. So this is an epistatic effect of the MCR1 gene. Now let's look at another example of epistasis in the fur coat color of Labrador dogs. So there is this gene which we denote by E, which we denote by B. First, let's talk about B here. If this gene is present as a capital B, the dominant, the wild type version, then it gives black fur, okay, irrespective of whether it's present in single dose or double dose. The recessive version of this gene, if you see two small B's, the dog would have a brown fur. Now, the effect of the black and the brown is externally controlled by, there's a second gene that is interfering with this expression, and that is the gene, the alleles of which are denoted by this letter E. So if this gene with the E's, if they are present in homozygous recessive form, that means two small E's, then this totally neutralizes the effect of this black and brown. So a dog that has two small E's, irrespective of whether it has a capital B or a small b, would end up having yellow fur. So this is another epistatic example of one gene masking the effect of other gene. Another example of epistasis is a very famous example called Bombay phenotype. Bombay is a city in India. That's where it was first discovered. Nowadays, that city is known as Mumbai. And this is also known as small h blood group. And this is because the blood grouping that we just learned, the H gene is epistatic to the I gene. What does this mean is, when that A or the B antigen that is sticking to the outside of our red blood cells, they need a particular H protein to adhere onto the surface of our red blood cells. If there is no H protein, which happens in the case a person gets two recessive, small h genotype, this is due to a mutation in a particular gene. So if there is no H protein, the A or the B antigens cannot get attached onto the surface of the red blood cell. That means if an individual has two H genotypes, they would always have, then even if the A antigen is right there, it cannot stick to the red blood cell. Even if the B antigen is, it cannot stick. So the individual would end up having a phenotype of the type O, blood type O. So this is another example of epistasis. So we've already seen so many deviations: incomplete dominance, codominance, epistasis, lethal alleles. Now there are two terms that are used in genetics that are used to solve genetic problems. They are called penetrance and expressivity. Penetrance means all or none expression. Okay, a gene will either express to its fullest ability or it will not express at all. So we see how penetrant the gene is. And then expressivity, on the other hand, is a range of expression. Okay, this is the severity or extent. So a genotype is incompletely penetrant if some individuals with that genotype, if they do not show the phenotype. For example, polydactyly means multiple fingers, more than the 5 fingers or more than the 5 toes. And it is seen that people with mutations in the genes associated with polydactyly, they have a wide range of expressions. Some individuals will have 6 fingers, some might have 7 fingers, some might have 8 fingers. So you see a range of expression, okay? And a phenotype is called variably expressive if the symptoms vary in intensity. It's the same thing. So sometimes these two terms kind of get blurred, and like, what are we talking about? Are we talking about penetrance, or are we talking about expressivity? Another deviation is pleiotropy. Pleiotropy means where one gene is controlling several functions. That means one gene is controlling more than one trait. So for example, in Labrador Retriever dogs, the gene that will control the color of the fur will also control the color of the nose, the lips, the eyes, etc. Some other disorders of metabolism like sickle cell anemia, phenylketonuria, albinism are also here. One gene might be controlling just not this one trait, but some other traits as well. A classic example is this disease called porphyria, which was common in European royal families. So the porphyrias, these diseases are caused by absence of or deficiency of enzymes that are needed to make heme. Heme means the iron part of your hemoglobin. Hemoglobin is present inside your red blood cells, and this is important to transport oxygen. So if you have deficiency of hemoglobin, a person is said to have anemia. And this making of the hemoglobin, making of the RBC requires several steps. And there are enzymes that are helping in these steps. So if these enzymes become defective, if one or more enzymes in this entire pathway of making the heme becomes defective, then some of the byproducts of this reaction, they start accumulating, and then they might be getting excreted out along with urine, or they start accumulating in your tissues. And actually, because hemoglobin is responsible for the red color of the blood, this is the red blood cell. So when some of the byproducts got accumulated, it led to people having reddish teeth. They were, very sensitive to light, and actually this is believed to have led to stories of vampire or werewolf, or because these people, royal families, they used to live in big castles, and they had this disease, so their teeth looked like as if they had just taken in blood. So you see here, these are what happens when the missing enzyme is accumulating and it is getting out through the urine, and the urine becomes dark red in color. If it is in the digestive system, a person would get constipation, abdominal pain. If it's in the muscles, nervous tissue, all of these different symptoms. Genetic heterogeneity. Heterogeneity means when mutations are occurring in different genes, but the effect is coming out to be the same. So they are producing identical phenotypes. For example, hearing loss, this is an effect, this is a trait or an abnormality. And this is being caused by 132, mutations or 132 versions of the gene. So 132 autosomal recessive forms. Or the brittle bone disease, osteogenesis imperfecta. This has at least 2 different genes involved in a variety of cases. There are 19 different types of osteogenesis imperfecta, or even Alzheimer's disease. 4 different genes could get mutated to produce the symptoms of Alzheimer's, okay? And why does this happen? Well, how come it is that different genes are getting mutated, but they're producing the same result? It could be, one of the possibilities is that all of these genes are actually working in the same biochemical pathway, okay? They are encoding, they are making those enzymes that are being used in the same biochemical pathway. And that is why when any single of these enzymes is not made or is defective, the same pathway is getting affected, and so the same product is not getting formed. Another type of deviation is called phenocopy, and phenocopy means a particular trait, a defect or a syndrome. It looks like it has been inherited, okay, is genetic, but actually it is caused by an environmental exposure. It's an environmental insult. For example, if a person gets exposed to cancer causing or DNA damaging agents, teratogens like thalidomide, it can cause limb defects in the newborn baby, and it might look like that the baby was a genetic disorder that was not formed, or even an infection. We all know AIDS, okay? AIDS means acquired immunodeficiency syndrome, and this passes through bodily fluids. So if a woman who's pregnant gets AIDS, the woman can pass it on to her unborn baby because there is fluid getting from the maternal blood circulation into the fetal one through the placenta, and the AIDS virus can be transmitted from the mom to the baby. And when the baby is born, if much was not known about AIDS, if science was not as developed, people would have thought that this is a transmitted disorder, that AIDS is a hereditary problem, but it is not. So knowing the entire human genome, now that the entire human genome has been decoded, it has revealed that actually deviations to Mendelian inheritance are a lot more common than what was originally believed. And so many things, epistasis, genetic heterogeneity, they are starting to overlap. For example, Marfan syndrome is a problem which is an autosomal dominant disorder. Here you will see individuals who are really tall, they are thin, long arms, okay? These are effects of genetic heterogeneity, epistasis, both, or interactions between genes can change the penetrance and the expressivity. For example, Huntington disease, which is also an autosomal dominant disorder. Now we come to something very interesting called mitochondrial disorders. Now, most of the times when we're talking about genes and hereditary diseases, we are focusing on genes which are present in the nucleus. So they are nuclear genes. But apart from the nucleus, there is this organelle in our cells that also contains DNA, and that is the mitochondria. And I think I mentioned when I was teaching you cells, that mitochondria were originally independent organisms, and they somehow got inside other cells and the symbiotic relationship developed. There are 37 mitochondrial genes. These genes are very different from nuclear genes. They are small in size, they are circular, but these genes also encode proteins, okay? So diseases can result from defective mitochondrial genes being passed on from the mother to the baby. Mitochondrial genes are only inherited from the mom because the cytoplasm comes only from the mom. The mitochondria are coming from the mom only, from the maternal side. From the dad's side, it's only the chromosomes that come, so only nuclear genes come. So some examples are mitochondrial myopathies, which are muscles that are weak, you're not able to work, you not use them efficiently. There's a particular optical atrophy, means vision loss, okay, impaired vision. And nowadays in research, we can use ooplasmic transfer technique. This is the technique that was used to create Dolly, the clone, the sheep clone. I think all of you know that. So this is where we can control what is getting transferred from the cytoplasm. And actually we can just selectively customize what are we taking from the egg, what are we taking from the ovum, and we can take the cytoplasmic material from a third cell. So let's understand some of the, at least one of this, the mitochondrial inheritance abnormalities called heteroplasmy. Hetero means, not the same, different. So there might be a time where a woman in her cells, all the mitochondria do not look the same, okay? So the mitochondria in the same cell are different looking. That is it's a rare situation, but this is the mitochondrial sequence is not the same in all copies. So some of the mitochondria may be healthy and normal, while some other mitochondria might actually be abnormal, might have mutations. Now, let's say this is the ovum containing some normal and some abnormal. So you see this cell, this is an oocyte or the ovum. The yellow colored ones are the mitochondria that are normal, healthy, it's without the mutation, wild type, while the purple ones here, not this one, this is the nucleus, the other ones, these are the mitochondria with a mutation that make it abnormal or disease causing. So when this particular egg fuses with a sperm, fertilization occurs. Now, depending on, which type or how many mitochondria are getting, if this particular egg has how many abnormal and how many normal. So depending on the number of abnormal mitochondria inherited by the fertilized egg, If this fertilized egg got only one abnormal mitochondria, no problem. Okay, it is within the tolerance level. Two abnormal, fine. Three abnormal, fine. But what if the fertilized egg got four? This is the threshold. It crossed the threshold. Now this offspring or this particular muscle cell of the offspring will start showing an abnormality. So as soon as the threshold is crossed and the offspring inherits more of the abnormal mitochondria, the offspring will show a particular disease, and it could have a wide range depending on how many abnormal mitochondrial genes the offspring has inherited. Now, the mitochondrial DNA helps in forensics. It helps experts to reveal history, to reveal ancestry from the maternal side. For example, during the French Revolution, when the king, Louis XVI, and his wife, they were removed from the throne and they were thrown in the prison. The king was executed. The wife was also, the queen was also later executed. And the prince, the heir to the French throne, he was in the prison for a couple of years. And then there were all these conspiracy theories, because some people said that the prince died because of a lot of diseases and malnutrition and no care. But there were some other theories as well, that the prince escaped and another child was put in his place and so on. So this mystery was solved several years later using mitochondrial DNA, that the remains of the child, after he had been buried, that some DNA, some mitochondrial DNA was extracted from this, the remains of this body, and then from relatives of the queen's side, and they even had some sample from the queen's hair, and they extracted mitochondrial DNA, and they could show, give concrete evidence that there was maternal inheritance involved. So that was indeed the prince who had died in the prison. Now, there are two common genetic phenomena that occur in all cells, and those are known as linkage and crossing over. And linkage and crossing over are also two things that give you deviations from Mendel's laws. In general, what we see, what Mendel had seen was, because in his second law of independent assortment, he had said that traits, the trait on one chromosome does not interfere with the trait on another chromosome. At that time, he did not know that they were on different chromosomes, but we know that. So he was lucky that the traits were located on different chromosomes, because if the traits were on the same chromosome, then he might have seen something different, because depending on how close two genes are on the same chromosome, the two genes might get linked, might form linkage. What this means is, let's say if this is the chromosome and these two genes are very closely located, they develop a liking for each other, okay? They share a bonding with each other, and then they don't want to get separated. They like to stay together. They like to stick together. That is known as linkage. So he would not have seen independent assortment. He would not have seen segregation, because the genes are closely located together on the same chromosome. So linked genes do not assort independently in meiosis. Rather, they like to get inherited together. So he would not have seen the typical Mendelian ratios. And what happens to those genes that are not closely located? The farther away two genes are from each other on a chromosome, then they don't like to stay together, and they actually, they might assort differently, and they show recombination. So chromosomes would recombine during crossing over. There's a particular phase in prophase 1, and this actually produces variety in sexual reproduction. So let's say these are two homologous chromosomes. You see the two genes here between the blue and the green in meiosis 1. These two chromosomes exchange some pieces. Okay, you see some little bit of the green came here, a little bit of the blue went there. And this produces variety in sexually reproducing organisms. And that is why you will see, even in human beings, although you might say this person has mom's eyes, dad's hair or whatever, but there's some things that would be very unique. Not exactly, it's not like, everything is a carbon copy of the mom and the dad. There's always some variety, something very unique to that individual, and that occurs because of recombination. So the alleles present in a dihybrid could have two different configurations: two dominant or two recessive alleles could be on, on the same chromosome, then that is a cis configuration, that is known as a cis configuration. While trans is when there is one dominant and one recessive allele on this one, while one dominant and the other recessive allele on this one. So this is a trans configuration. Now, what is the frequency of recombination and frequency of linkage? Remember, linkage and recombination are opposite to each other. Genes A and B are far apart. So if they are far apart, they don't want to stay linked. Then there would be a higher chance of crossing over. While genes B and C, if they are close together, then they love each other, then there is more likely that they will stay together. So they will be linked. And this property of genes in chromosomes is used to create maps. They are known as linkage maps, okay? And it is said that the frequency of recombination between two genes is proportional to the distance between them. That means this is a linkage map. If I say 1% recombination, that means those two genes are one map unit apart, and that one map unit is 1 centimorgan. So let's say if I say there are these three genes, X, Y, and Z, and I give you the percent recombination between gene pairs. The percentage recombination between X and Y is 10%, between X and Z is 4, and between Z and Y is 6. Now draw a linkage map. So from these figures, it's clear that X and Y you draw here and you put 10 map units in between them. What about between X and Z? So it is only 4. So you put Z here and this is 4 map units. And then between Z and Y, it is 6. So this is 6 map units. Now, just by looking at this map, X, Z, and Y, if I ask you to see this map and predict which two genes would more likely be linked, of course, it's X and Z. They are very close to each other. What are the two genes that are most likely to show crossing over, to recombine? X and Y, they are farthest apart. So the genes that are farthest apart from each other, they will cross over, while the genes that are close to each other will stay linked. And this property is used to create maps. This property is used for high throughput studies. When we are studying populations, when you predict something about human population, you just cannot study 4 or 5 individuals and give a generalized conclusion. When we do population studies, we need thousands of samples. So we need to know some genetic foundation laws. We need to know what is linkage disequilibrium, LD. Linkage disequilibrium, this is the definition. This is the non-random association between DNA sequences. That means how close two genes means DNA sequences are each other. The closer they are, the more likely to be they are linked, and the more likely they are going to be inherited together. And the entire human genome, you will see there are LD blocks. So after the human genome has been decoded, we know what are the genes that tend to stay together, to stick together, and are inherited in offsprings in one block, in one LD block. While these two genes, because they are so farther apart from each other, they don't tend to be inherited together in one block. Instead, there would be crossing over. And this property is used to solve linkage problems. For example, there's a problem here, and it's talking about two particular autosomal dominant traits. So let's say the genes for autosomal dominant trait nail-patella syndrome. Nail-patella syndrome means a disease where the nails are not formed, and the patella, that means the kneecap, is also defective. So let's say the letter N, autosomal dominant, remember, that means a single dose would produce the trait. And the ABO blood type, these are 10 map units apart on chromosome number 9. So the problem is this is a couple, Greg and Susan. They each have nail-patella syndrome. That means they both have this autosomal dominant disorder. So once you already know this information, that means you know that they have one capital N, right? They both have nail-patella. Now Greg has type A blood, while Susan has type B blood. And they go to a genetic counselor and they ask the genetic counselor, we want to get married, but we want to know what is the probability that our child would have normal nail and knees. That means our child would not have the nail-patella syndrome, and would have a particular blood type, type O blood, because they have A and B. Remember, A and B blood type, if it is heterozygous A and heterozygous B, then they can have this O blood type. Now the genetic counselor, to help them understand it, the genetic counselor first has to know the family history of both Greg and Susan. So he starts asking, Greg and Susan, who among your parents have Nail patella syndrome, because it is autosomal dominant. So every generation there would be an expression. So he gets all the information about the couple's parents. He gets the blood grouping done from the couple's parents. And this is what the genetic counselor comes up with. This is for Greg, this is for Susan. Both of them have Nail patella. Greg has blood type A, Susan has blood type B. And then asking them about their parents, the genetic counselor is also able to draw this, that Greg is getting, his mom has Nail patella and she has blood type A, while his dad is normal, healthy, does not have Nail patella, and he's a blood type O. So Greg's alleles are in cis configuration, both dominants on one side, recessives on the other. While Susan's alleles are in trans configuration, because the mom, let's say, has Nail patella, but she has type O blood, while the dad is healthy, but type B blood there. And based on these, the genetic counselor comes up with all the different options for what the sperm could look like and what the oocytes could look like. And then using the recombinant map calculations, these are the frequencies that are given. That if there is a paternal, recombination, paternal frequency of these gametes would be this, while recombinants is this. Now, in order for Greg and Susan to have an offspring who does not have Nail patella and who's a blood type O, the probability is calculated by multiplying this 0.45 times the 0.5. That means 5% and 45%. That is 0.0225 or a 2.25% only. So this is how a genetic counselor uses information from your pedigree, from the person's genetic history, and can come up with a probability of a particular trait in the offspring. Now, genetic markers are common terms that we use in GWAS, that means genome-wide association studies, and genetic markers are like landmark sequences, DNA sequences, which are near your genes of interest. So we might be talking about a particular gene, and then in order for other people to understand which gene we are talking about, we talk about a very famous gene, a landmark gene that is close to it. So we say chromosome number this and this many bases upstream or downstream of, let's say, P53 or some important gene. And this helps, and these are used in, as I said, high throughput, big studies involving thousands and thousands of subjects, genome-wide association studies. And in these genome-wide association studies, you cannot just go by individual genes. You have to talk about blocks of genes. So we look at LOD or LOD score, which is known as logarithm of odds score, as well as haplotype blocks. So the logarithm of odds or LOD scores indicates the tightness of linkage. That means what is the probability that this particular landmark gene, the marker gene, and the gene of interest are going to be inherited together, how close they are located. So in general, if the LOD score between your gene of interest and a marker gene is 3 or higher, then it would signify that these genes would be inherited together. And when we do these studies, we use haplotype blocks. Haplotype blocks, the entire human genome can be divided into blocks of genes. Those are haplotype blocks. And in each haplotype block, we have a tag landmark, a tag SNP. And as long as our gene of interest falls in that particular block, we really don't have to individually study each gene. We will take that block and take one of those tag marker genes, and we can predict about the other genes in that block based on the property of that tag marker gene. Okay, property means inheritance property. So let's say a particular block has 20 genes. Okay, and those genes will try to function in the same way, try to be inherited in the same way. And we have two landmark genes there. We call them tag SNPs. So we just have to know about those two tag SNPs, and then we can, sufficiently confidently uh, uh, hypothesize about or predict about all of those 20 genes based on the properties of those two genes. I'm trying to help you understand something difficult in an easy manner. This is what haplotype blocks do. So haplotypes, remember, are a group of genes. And then in that group, we know these genes are going to be inherited together, and we just have to select one or two landmark genes. And then those landmark genes would help you or help the readers know about all of those 20 genes in that particular haplotype block. And we are done with the lecture. Do you have any questions? No questions? Okay, give me, if you understood this class, give me thumbs up. If it was difficult, let me know, I can understand. Thank you. Do not forget that you have your homework 1 and other assignments. Do not miss the deadlines, because those are the easy points to grab. So turn in all your assignments on time so that you keep getting those points. Okay. And now we can end the class for today. I'll see you tomorrow.